So today I will talk about super resolution microscopy. And you can see here you have, in this graph, you have a whole range of fluorescence microscopy techniques. They allow you to image across various biological scales. Now I have to mention that when using collecting light to observe a sample, biological sample, you have a hard resolution limit. And this limit is about 200, 250 nanometers. And this is due to the physical property of light and also the limits of the imaging system. So the microscope and the lens can only collect a certain amount of light, which encodes all the details in your image. So what super resolution microscopy does, it was methodology that were developed to break this diffraction limit. And today I will present three main super resolution techniques. So structured illumination microscopy, or SIM, stimulated emission depletion, or STED, and single molecule localization microscopy techniques, or storm. So those are like three techniques that we have available in the light microscopy, and they go beyond that diffraction limit. So that resolution limit, just to introduce it here, is that hard resolution is 200, 250 nanometers, which means that if you have two objects that are 200 or 250 nanometer apart or more, you can distinguish them in the microscope and say I have two objects. But anything that is below, if you try to observe it directly with regular microscopy, you just see a big blurry blob, and you cannot know if they are 100 nanometers apart, for example. You cannot know that you have two objects. You just see a big blurry blob. Okay, so to come back on the diffraction limit, and what it means and why it's there when you do fluorescence microscopy. If I take, for example, the fluorophore that emits light, they will emit diffract light in every directions, and the microscope will collect this light. And so the image that you see in the microscope, you end up, instead of seeing just one point that it's 1 nanometers as the object you want to image, you have a diffraction pattern, and that it's called the PSF. So this is the pattern you see in your microscope, the image. So you have a characteristic shape with a high intensity in the middle, and those diffraction rings that forms the image. And so this is wider. So this is not 1 nanometer, this is expanded. And this is how it looks like on the XY plane, and on the Z plane. This is also how it looks like. So this is what is called a point spread function. So if you model this in intensity, you see that characteristic peak of intensity. It forms a Gaussian. That's what it's called, the airy disk here. And you have minimas that correspond to those rings on the X plane, for example. And so the resolution here, if I have two objects, this is how I see objects on the microscope. I have two objects that are close to each other, but within the resolution limit, I can identify them, and I can say I have two objects. When they get very close to each other, they start merging, and here you can't distinguish anymore. And so there's various criteria to define this resolution limit. So you have the Rayleigh, the Abbe, and the Sparrow. And when you reach the limit here, you still see two peaks, and you can say maybe I have two. I know that I have like two different objects close to each other. But if you go below, then they start to merge together, and you just have a big PSF, and you don't know if you have one object or multiple objects are encoded in there. Those Rayleigh, Abbe, and Sparrow criteria describe resolution. And here I will concentrate a little bit more on the Abbe, because that's the one that is more mainly used for microscopy, to define resolution in microscopy. So in the best condition, this is the resolution you can reach with light microscopy. So when you have a system that can collect as much light as you can, so typically a lens, an immersion lens, you can reach a lateral resolution of 200, 250 nanometers. and in the z-axis around 500 to 700 nanometers. So this is how Abe was describing resolution. So that's his formula when you see that the resolution is dependent on the wavelength and the numerical aperture. So it's lambda over 2Na. So the wavelength is the wavelength of light you're using, and the Na is reflects the capability of the lens to collect light. And that can, so that's that N sinus alpha, which is the angles the light can be collected, and the refractive index. And so typically the lambda, the wavelength of light, is between 400 and 500 nanometers. And the numerical aperture that we have available for light microscopy is around 1. So a little bit below 1. The numerical aperture is a little bit below 1 for air objective, and a little bit higher, up to 1.45 for oil objective or immersion. Yes. So for a very long time, it was that resolution limit for light microscopy was believed to not being so like that was like a hard resolution limit, so you could not overcome that limit. But super resolution were developed to break that diffraction limit, and that's with imaging methods, and also taking advantage of physical property of dyes. So by the combination of the two, they were able to break that hard limit in resolution. And so the people who allowed the development of those techniques were even rewarded Nobel Prizes, a Nobel Prize for it. So now I'll start explaining the free super resolution technique that we have available here. And broadly, any light microscopy super resolution technique can fit within those three categories. So structured illumination microscopy, when you use a pattern to resolve details that you would not normally see in an image. Stimulated emission depletion or STED microscopy, when you engineer the PSF and you narrow the excitation region you're imaging. And finally, single molecule localization microscopy, when you sequentially image fluorophores and reconstruct an image with higher resolution. So first, structured illumination microscopy, or SIM, when you have, so the technique uses a known illumination pattern that is formed with a grid in the light path. And as you excite the sample, instead of having an homogeneous illumination, you have that illumination pattern that goes on top of it. And that creates interference that generates a third type of pattern that can be measured. And with that non-illumination pattern, those generated pattern, you can resolve structure that you would not normally see. And so to get a bit more into detail on explaining this concept, first I have to explain how the light is collected by the lens. So when you have an object, so like a sample here, that emits light, it emits lights in every direction. So you have fluorescence that goes in every direction, which you can see it as rays of light. And so angles, it goes in every angles. Now, when you have a lens, it goes on top of your sample. And that lens only have a certain capacity in picking that light. So it doesn't collect absolutely everything. It just collects a portion of it, what it's capable of seeing, basically. And that means, unfortunately, so those rays that are not going into the microscope, you can't see them, but they also contain very fine structures in your image. That means that don't even go in the microscope. So you lose in resolution because it's not even seen by the microscope. It's not seen, but if you use a pattern that you know, which is the illumination pattern that SIM microscopy is creating onto your sample, you create interaction with the details in your sample that you don't see in the lens. And that third pattern indirectly gives you that information that don't go in the microscope. And this is, that third pattern that I mentioned now 3 times is called the Maury fringes. So it's exploiting that phenomenon, that interaction of light between 2 structures pattern. And so a way to describe it, if you ever try to take a picture of your PC screen with your phone, you start having unwanted patterns that gets generated in your image in your phone. And so this is a similar process, but in this case, we want it because they reflect frequency or light that is not collected by the lens. And so, yeah, so then it's a simple equation. So you have a sample, you don't know all the structures in it because the microscope can't take everything, but you add that pattern, that illumination pattern. So the illumination pattern that is known is a stripe of light. So you have dark areas and light areas that goes onto your sample. So you know that, you generate those third wanted pattern because it interacts with the sample. And so you can deduct the information into your image. And so yes, so that means also because you have that illumination pattern, you're not illuminating all of your sample at once. You have that pattern that gives you a strip of light. So when you acquire an image, typically you have to shift that grid. So you shift the pattern so that if here it's not illuminated, then when you shift it, it gets illuminated. And same, this region that was not illuminated, that gets illuminated. So you do 3 shifts like this that are also called phase. And that allows you to cover your entire sample, but that's covering in one direction. The Maury fringes are generated by seeing organization in your sample. And so to cover the entire sample, you also do rotations of that pattern. And the combination of this can be computed to resolve the structure of your image. So that's the SIM processing. So after you acquire the SIM, that also means, I forgot to say, that also means that you acquire 9 to 25 images per plane you want to image. So it's quite a lot, and you are at risk of bleaching or something. So that's something to be, yeah, to look at. And also if in the time of so the acquisition takes longer time. So if the sample moves, or if you have a live samples that move, you also lose the information. Now for the processing, to explain the processing is happening in the frequency space. And to explain what that means, I need first to precise that an image can be described in different manners. So there's different ways to describe an image. The more intuitive way to see an image is that it's a series of pixels. So you have an image in the spatial domain, it's a grid of pixels, and each pixels have an intensity. And the entire, like the series of all those pixels from the image that we can see with our eyes. Now, if, for example, you use ImageJ and you want to plot, to do a line plot, for example, I take one line of this image, I do a line plot, I end up with an intensity profile. And for example, for one line, I can say my intensity goes up and down. So I transform this image from a series of pixels into a graph. But that's the same, I'm describing still the same data. And so what the Fourier transform does is I transform an image, a series of pixels, into a series of frequencies. So it's just the same way to describe, a different way to describe the same image. And that's interesting because in the frequency domain, when you have coarse structures or big structures, they are in the low frequency, so close to that bright dots. Everything that is close to that bright dots are like big structure in your image, and fine details are represented by the frequency that are more peripheral. And so coming back to that schematics on how a lens collects light, there's a part of it doesn't collect everything. So all the small, the fine details are actually high frequency. And so the microscope acts as a mask. It doesn't see all the frequency in your sample. And that is represented by the diffraction limit. So this is a diffraction limit in the diffraction limited image in the Fourier space. And the whole point of super resolution is to try to access that information, those small details in your image. And yeah, in the frequency space, there are frequencies that are in the black region here. And so that's why the same does with the Fourier, with the phase shift and the rotation. So the Maury fringes that are generated are seeing details that are a bit more spaced out in the Fourier space. So the more the details, it sees the details. And so it can be in the Fourier space, they can be reattributed in the right region. And with the rotation, you just generate a bigger circle. So from what you were able to see with a normal confocal, you start having information that are outside that the microscope were not able to directly measure. And yes, with the rotation, you just cover more. And so in the Fourier space, you have more frequency, which means more details. And this is a way to describe your data. So with inverse Fourier transform, you can just change that series of frequency that describe your image into a pixel, a series of pixels that describe your image again, but in a more visually appreciable way. So that's how SIM works. We have one here. So that's the Zeiss LRF for some of you who maybe already used it. So you can see the specs here. And the resolution is still dependent on the wavelengths. So if you use blue and green dyes, you will have a better resolution than what red and far dyes. So that applies with, so the ABI formula still work in that regard. Still applies. Yeah, so here's some images to just show you two-color SIM imaging. When you have here actin and mitochondria, so you can appreciate the difference in details you see in your image. So you go from wide field to SIM here, just have a sharpened image, you start identifying a bit better the fibers. You have an improved resolution. Same here, where, so if, for example, you just want to count the blue structure, which were bacteria, SIM won't bring you much more. But if you start looking at very small objects, like for example, on the top, when you have that blue and red overlap, when you go to SIM, you start seeing that they are not one on top of each other. So you start with small objects, seeing more information, and you resolve more of your structures. You have here, yes, so those are cells infected with salmonella, and you can see that there's some structures that surround them. Now, the next super resolution technique that works in a very different way, which is stimulated emission STED microscopy. And for this technique, you engineer the PSF, so you narrow your emission signal to have a precision localization. So you have to see this method as a classic, first as a point laser scanning microscope, like a classic confocal. But on top of your excitation beam, you have a STED beam, which is a donut that is shine, completely superimposed with your excitation beam. And so that donut have a middle that you can see here, which is a complete zero. So you have a complete zero in the middle, and the STED beam is able to switch off the molecules that are on top of it. So you create local contrasts, that's the way I say it, but you have switch off molecule all around and a spot in the middle when you can collect the fluorescence from there. So that narrows what you're observing. But more importantly, how does the STED switch off the fluorescence? So when it comes down to that path of excitation and fluorescence, so when you excite a molecule, it goes to the excited state. And when it sees the STED donut, instead of emitting fluorescence in a regular way and creating those green photons, for example, here, you just force that excited molecule to emit at the same wavelength as donuts. So it's not a regular fluorescence emission. You're just pushing an emission of a photon that the microscope don't see, because the STED donut has a higher wavelength than your emission and your emission spot. So you are removing basically that tail of fluorophores, and you only observe the fluorescence at the middle of this spot. And with this technique, the PSF is not as wide as it is by default, and you can reach a resolution down to 20 nanometers. But this process is also dependent on the intensity of the donut. So the highest the intensity, the better the resolution. So you switch off the molecule all around, and you can see it here visually. So if you have a low intensity donut, your middle spot is bigger. But when you increase the intensity, you narrow your region, so you zero in the middle. And you can see it here with the equation of resolution. So you have that normal array lambda to NA, but you have another term that is added to it, which is proportional to the intensity of the donut. So IS here represent the saturation. So you can saturate how much you deplete in the region. So here the saturation intensity IS represents 50% of the fluorophores. So so with this technique comes also a risk of bleaching, because you have an excitation beam which is quite wide and a donut that comes on top of it, but you're only imaging in a microscope that region in the middle. So that means that to get this higher resolution, you have to do a much thinner raster scan. So you have to scan more, and the time you get to another point, actually already excited your sample multiple times until you reach that spot in your image. So now there's very performant dyes that don't bleach as fast, but so the sample is seeing multiple time your excitation beam until it reaches the spot you're imaging. So that's to take into equation. Now the same process applies for 3D STED. When you have a beam that generates that donuts for the XY plane, and the beam is split to also generate caps on X on the Z. So you will have a cap on the bottom and a cap on the top and on the bottom. And so you encapsulate the region with a 3D donut. You have a donut and two caps on the Z axis. And so that also narrows the PSF on the Z plane, so that you also see with better resolution. So you also get an improved resolution of, you can reach 120 nanometers. So you are below the diffraction limit. Thanks. So let's switch off the intensity. And so we have also a STED microscope in the facility that is available to use, and you can see the STED laser. So we have different STED laser: 592, 660, and 7775 to be able to image green fluorescence, red and far-red fluorescence. And so as I was explaining, the STED laser is always higher in the spectrum than your emission, than the emission you're looking at. And now here, again, to have a visual of the different techniques the images you get with this technique. So here you have the tubulin that it's labeled with antibodies, so primary and secondary antibodies. And you can see on the confocal, for example, if I look at the top structure, you have two fibers. So you can resolve that you have two structures that seems to cross. And when you go to STEDs, you see that those two fibers are actually 4 or 5 fibers. So you get down to really see details in your image. Yes, so if I do a line plot here of the intensity with the green image, so the one that's with the confocal, I see 2 peaks. That shows me that I have maybe 2 structures here, 2 fibers here. And when you're in a STED, you can also represent the data in a way that you see that you have multiple peaks. So you resolve this structure. So on a confocal, you're limited by the diffraction limit. So your resolution is about 250 nanometers. And when you go down to the STED, at least for this image, you are around 90 nanometers. But the actual diameter is 25 nanometers. And you have to take into account here the presence of antibodies. Because to be able to see the structure, you need fluorescence. And to have fluorescence, you use antibodies. And the antibodies are structures that have also, they're about 10 nanometers. So they also take up space. Second image, so here you have neurons that are imaged, and so same principle. You can see that here you have the confocal images of your colors. So we're looking here at synapse. And when you use the STEDs, you start seeing with much more details how your 2 elements localize in your image, and that they have different structure. They look quite similar. Here you can see that one is more dotty, the other one has more intense structure. Same here. So you see the difference between confocal and STED, and you can clearly see an improvement in resolution. And so that one is looking at recycling endosomes. And so you have here a confocal image. So this is 3 colors. Then if I zoom in, this is what you get with a confocal microscope. And when you switch to STED, you start seeing a bit more how the different proteins are localized. Are they co-localized? Are they next to each other? You also start seeing, you also start seeing structures that you wouldn't see before. So like on the top left, there are some dots that basically appears. They get a bit more resolved with STED. And now the last type of microscopy, of super resolution microscopy, which is single molecule localization microscopy with palm and storm. So this technique that uses the fluorophoto chemical properties. So it's exploiting a little bit those properties to be able to resolve your image. And so to describe how it works, if you have an image with a lot of fluorophores that are very close to each other and you can't resolve them, they just appear as a big blob and you don't know how many are there. The idea is to be able to switch them in and to switch them on individually and image them one after another. So if you're capable of imaging them one after another and identify the position for each of those fluorophores, then you can reconstruct an image. So this is a sequential imaging. It takes quite a lot of time because the idea is to isolate and have a sparse sample. If your sample is sparse, you can identify single molecule, and from there you can recombine and recreate your image. And so yeah, with this technique, you can reach a resolution that is down to 10 nanometers, but this is also limited by your labeling. So you need to have a good labeling to be able to have all your structure back, basically. So being able to localize, so if you are able to image one spot, one single molecule spot, and you want to localize its position in X and Y, this process is like photon dependent. So this is what this slide is showing you, is that the localization precision is related to 1 square to 1 over the root of the photon count. So the more you have photon, basically, the easier it is to identify the centroid of the fluorophore that is imaged and to define its position. And you can also see that if you have background in your image, that will also make the spatial resolution a bit worse. So the graph here is showing the precision, the localization precision, the errors against the total of photon detected. And the more you have photons, the better you get at pinpointing where is the center and where is your molecule. Yeah, so similar here. So this is how you image your spot in your, so this is what you see in your camera. You have your diffraction limited spot in an array. And by being able to process where is the center, so you do a Gaussian fit, and you have enough photons to identify where is your center, you recreate an image with a thinner grid and a more precise position. And so how this is possible, because the idea is to be able to image one molecule after another. So not exciting your sample and having all the molecules being light up at the same time, but more one after another, you want to see molecules. And so you have different, and that comes from the properties of the fluorescent that you will use in your image. You will use in your sample to do this. So you have different type of, this is palm microscopy, where you have different type of fluorophores that allows you to do, to activate molecules that you can see sequentially. So you have the photoactivable fluorescent proteins, where they are all off to begin with. And if you use a specific wavelengths, you can little by little activate them. So you do sequentially. So you typically acquire over 1,000, if not 10,000 images to sequentially see the fluorophores that compose your image. And by doing this, you can then reconstitute how your image looks like. And then you have also photoconvertible fluorescent protein, which are proteins that are, for example, here all green, but have the capability to be switched into another color. So by using a specific wavelength, you convert those green fluorophores into a red spot that will be sparse in your object. So little by little, again, you acquire them, and that allows you to reconstitute your image. So with single molecule localization microscopy, the key is to have a sparse sample. And for stochastic, so then, the last type of single molecule localization microscopy is storm microscopy, or stochastic optical reconstruction microscopy, where here the fluorophores that are used have the ability to be switched on and off. So you have fluorophores that are in the on state, but have the ability to go into a long lasting dark states. So they're not bleached, they just don't emit anymore, they are in this state. And stochastically, they can go back into the ground state for excitation. And that process can be pushed or forced by oxidation or using a blue laser, for example. So they will just, so you excite them, they go into a dark state, and they stay there, so you create sparseness in your sample. And then you can re-excite them, they go in the ground state, they get excited, so and so on. So you can acquire a serial image. So here, I'm just showing you how it works. So they can appear as blinking, because they go back in the ground state, get exciting again. So here it's shining a blue laser to reactivate again. So you image, you reactivate, they go into the dark state, that creates a sparse sample with just few fluorophores that are on, and so on. And so typically you acquire 10,000 images or more to cover your entire structure. So we have in the facility, we have the nano imager, that do storm microscopy. And yeah, so this is presented as just one box, basically, yeah, it has all the components to do storm microscopy. So this is available, and just to show it, to introduce you, typically when you do storm microscopy, you also use a turf or halo, so to be able to look at a very narrow region and identify fluorescent spots with a highest contrast. So how turf works, you have so you have an excitation beam that arrives in an angle and generates an evanescent wave of about 100 nanometers onto your above the cover slip. So you just see a narrow region with highest contrast, so a better chance to have localization precision on your spots. So here, a few images that show the differences. So you have here the storm images, so you can see how you resolve way better the spots. Yeah, so here it's just zooming in, and you start seeing more of the structures of those calveoli that marks the endocytosis of molecules in your cells. You can see here with more details. I'm going a bit fast through it. So, then you have here one power storm. So if you go from the white field to the storm, you can see that you start seeing way more structures and fibers in storm. Again, this is a reconstructed images, so progressively it was acquiring. So yeah, you go from an image that had a resolution of about 350 nanometers to when you're in storm, 70 nanometers. And now just a quick word on a 3D storm. I'm just going to load everything here. If you put in the path a cylindrical lens, you can introduce astigmatism in your image. And what that will do is that your PSF will be different depending on will appear different depending on the Z. So you have distortion if you're below the focal plane in one orientation, and distortion if you're above the focal plane in the other direction. And because of this, you're able to see those deformations into your image, and you can identify if, for example, fibers are above or below each other. So you have an information of the Z based on those aberrations that you introduce in your image. And yeah, so this is just the last example of how you can resolve structure with storm microscopy. So here you have a diffracted spot with regular microscopy, where you can see that you have the structure that could easily be interpreted as overlapping each other if you are within the diffraction limit. But when you use super resolution microscopy, you can resolve those two molecules in your image, and you can see that they're not overlapping, but they're more like next to each other. Oh, yeah, again, here to just mention that the labeling density is also a key parameter when you do storm microscopy. Is if you don't have enough labeled molecule, fluorescent molecule attached to your sample, then you don't resolve your structure anymore. But the more you have, the more intense the labeling is, the more you can resolve all the structures. So this is a bit what that slide shows. You create gaps if you don't have enough molecules tagging to your structure, which is less problematic when you use confocal microscopy or like a regular microscopy. But for super resolution, you need to have enough molecules so that you can reconstitute the structure. And this is also linked to the point I made before. It's when you have antibodies, they take up space. So the details that you're looking at might be overestimated in the length you're measuring. So you could use nanobodies. That links a little bit to Thomas' talk with labeling. So if you use different type of antibodies, you might improve that overestimation of size for your object. And that's it. So that's just a summary of the different methods I presented, and they are organized in the different resolution you acquire. So if you have SIM microscopy, you gain a twofold in resolution with the technique I presented. So if the diffraction limit is 200 to 250, the lateral resolution is 100 to 130. For STED, you go a bit below that in terms of resolution. So you can resolve structure around 25 to 80 nanometers, and quite similarly for localization microscopy. And so just here some take-home message on how to also make sure that you reach this level of resolution. So if you use cover slips that are to the right thickness, so 1.5 generally, you avoid spherical aberration. So that allows you to also get a correct image. Same if you want to remove autofluorescence. If you use phenol-free cultures, you have less fluorescence, so you can identify also a bit better your structure because you don't have extra noise. And same with having a mounting media with a higher refractive index, you improve the resolution. And immunostaining and the labeling can be quite important for super resolution technique if you want to be able to see, for example, your entire fiber. If you don't have antibodies everywhere, then you don't see, you have gaps. But if it's attached everywhere, then you start seeing it completely. And last slide is the acknowledgement. So the Light Microscopy Facility, the whole team. And for the data and the slide content, John, Nick, Anes, Jack, Sophie, and Kalina that allowed me to have those slides for this presentation.